Hello, everybody. Thanks for joining us today. It's about 11 o'clock here in Madison, so let's get started. I'm here today with Brian Anderson, who will be going over some of the functionality of DNASTAR's laser gene cloning and primer design capabilities. You may have noticed that your phones have been muted. However, we do encourage you to ask questions along the way. Just to ask a question, simply type it into the chat dialog and select Send to Host. I will then direct these questions to Brian to be answered for the whole group. If you need any assistance during the webinar or have any questions, feel free to send a chat message to me, sending that chat to host. Email me at webinars@dnastar.com or tweet us at the Twitter handle @DNAStarInc. And with that, I'm going to turn it over to Brian today.
Thank you, Jackie. Hello, everyone. As Jackie mentioned, my name is Brian Anderson. I am one of the technical sales scientists here at DNASTAR. I've been with DNASTAR for just under 8 years. Before coming to DNASTAR, I did have the opportunity to work at a company, whose primary products were utilized for protein expression and purification. So I do have some background knowledge and experience, definitely on the topic that we'll be discussing today. To start, I have a very brief PowerPoint presentation that will go through some of the points that we'll be covering today, and then we will go directly into the software to go through the logical workflows. So essentially there's 4 topics that we will be going through today. The first thing I'm going to do is introduce you to some of the tools that you can use for cloning, expression, primer design, and so forth. Primarily the DNASTAR Navigator, Seq Builder, and our Protein 3D application. We will bring in a sequence from Entree, and then we will PCR amplify a target. We will insert that target into a T7 expression vector. And in analyzing that express clone, we will translate that clone and also analyze the protein. So to start, primarily the 3 tools that we will focus on most of today will be our DNASTAR Navigator. This is a very helpful interface to directly link you to the various tools that DNASTAR can provide, as well as some help resources. Our Seq Builder application. Seq Builder is a very powerful tool for cloning, annotation. What I'm showing here in the background is the ability to change your visualization to whatever is dependent on your project. And then one of our newer applications, our Protein 3D tool, that we've just recently added our NovaFold algorithm that allows you to essentially bring in protein data bank projects, files, as well as bring in a protein and render or thread an image to create a 3D visualization using the Amazon Web Services. So these are the 3 tools that we'll focus on today. The target that we'll be looking at is a tissue factor precursor. So this is a protein that plays a primary role in hemostasis and thrombosis. This protein essentially has 3 domains. There's an extracellular, a transmembrane domain, and then an intracellular domain. We're going to focus on the extracellular domain, specifically an open reading frame or an intron that's present in that extracellular domain, and insert that into an expression vector. The session ID that we'll be focusing on is JO2846, and that's the part of the extracellular. The expression vector that we're going to work with today is an EMD millipore expression vector, PET39 specifically, 39B. This vector has a unique DSBA tag that's present on the 5' end of the multiple cloning sites. This tag is very helpful to export that expressed product into the paraplasmic splice. That will help obviously maintain activity, minimize degradation, kind of help with folding as well. So when you purify this, you're getting in the soluble, obviously, instead of the insoluble fraction and so forth. So there's also a 5' thrombin site where basically the end result, if I use the SCA1 and the XH1 cut sites, is basically a near native protein that when I cut it off the column, that all I have is a glycine to serine residue at the 5' end. So when we PCR amplify a target, essentially it starts with bringing that project in. So we're going to import that tissue factor precursor via Entree. I'll also show you how to bring in, simply drag and drop into Seq Builder as well. We're going to design primers to amplify that target into Seq Builder. We're also going to create design primers to add those SCA1 and XH1 enzymes at the 5' and 3' ends of that target to allow it to be cloned into that expression vector multiple cloning site. And then as part of this, we'll also optimize, the primary characteristics, optimize hairpin formation, dimer formation, and so forth. When we insert that target into the expression vector, we will basically utilize the built-in tools that are present in Seq Builder. We're going to focus on today the traditional cloning tools. So there's going to be a step where we're going to prepare the target, copy the target, and then essentially clone that target into that expression vector. We're going to look at the reading frame of that clone to make sure everything is in frame with the initiating methionine of the actual target, as well as the initiating methionine of the multiple cloning site. And then we're going to translate that clone into a protein for further downstream analysis, so we can look at rare codon usage. Maybe we may identify some rare codons that we need to express then in, say, a competent cell that has tRNAs for rare codons. The end result is an express clone. So this is an example showing that entire precursor that's been inserted into that PET39B multiple cloning site. As you can see, the parental information from the vector as well as the information from the target is carried over and applied and annotated to that express clone. And then we will also analyze that protein. So we'll take a look at the codon usage. We can apply some physiochemical methods against that protein. So for example, do a Kite-Doolittle hydropathy or hydrophilicity analyzation and verify, okay, is this protein more hydrophilic? There's a potential that it may be in the insoluble fraction or in the insoluble fraction. And then the end result is I can kind of show you how to basically take that protein, highlight any portions of that protein, and start folding it and generating a three-dimensional image so for further downstream analysis. With that in mind, let's actually get into the software. So I'm going to minimize here. And then I'm going to open the DNASTAR Navigator. So the DNASTAR Navigator here I have pinned to the my taskbar. You can also access this information directly from your, I'm on a Windows computer, so if you go to All Programs Back, Windows 7, navigate to the actual folders that we're using here. So DNASTAR Navigator 11 or DNASTAR LaserGene 11 Core Suite. But basically this is a very helpful utility in which you can directly access the various applications. As I highlight over a specific icon, as you can see in this specific case, Seq Builder, it gives me a very general background of what that tool specifically does. And there's also other helpful links to FAQs, tutorials. So for example, if I click on Seq Builder tutorials here, it actually directly leads me to DNASTAR's website where I can get more information and tutorials about that specific module that I'm working with. While we're on the website, let me just point you to a very helpful resource. From our support tab, we have a number of different resources that can further help you evaluate the software, look at the software. There's a number of videos, webinars, tutorials. If you're working with our Seqment engine application, there's template packages that you can look at, tech specs, and so forth. We put a lot of effort to supporting our software, and this is an example of that where you go to our website. I think there's over 150 videos that you can browse through based on functionality. So while we're here, I definitely want to point you to that support capabilities. All right. Another very helpful resource built into this navigator is also a help search function. So for example, if I click on this little exclamation point and go to search, what I can basically do is directly search all the search indices that are present in the individual modules of the software as well as our website. So for example, if I type in cloning here, as you can see, there's a number of search hits that come up. So if I click on TA cloning, today we're going to primarily focus on traditional cloning, but say you are in a lab that does TA cloning on a regular basis. What the software will do here, what this search function allows you to do, is it will actually walk you through the project where how to select a sequence for cloning, how to cloning that selected sequence insert, how to create the TA cloning insert, so it actually shows visualizations and walks you through the exact project, and how to end, the end result is saving that project for further downstream use. So this is a very helpful interface that we, within the last couple of versions, we've added to the software. So this is a very helpful tool that will get you to know the software if you're a new user, or maybe if you're a long-term user, teach you something that you may not know, didn't know exists in the application. So what we're going to do is simply we're going to launch our Seq Builder application. So as you can see here, I simply clicked on Seq Builder, and the Seq Builder window has popped up. So when you initially open the software, there's a blank window in which you can add data to. So one way that you can add data is simply just dragging and dropping. So on I have a left-hand monitor here. I'm simply dragging and dropping a .sequence file to the project. Or another option is I can also automatically input data from Entree. So from the File pull-down, I can select Open Entree Sequence. What the software is doing here is directly contacting Entree from NCBI. And then I can simply type in the session number. So in this specific case, it's J02846. And specify the specific database you want to pull this data from. So I'm going to select nucleotide, select OK. What the software will do is ask you to download and save that file in a specific location on your computer. So let's just save it to my desktop. And it will automatically open that file directly in the application. So an advantage of pulling information down from Entree is, very clearly if you go to the comment field, there's a number, some helpful pieces of information. So this is an older file, and will actually show you any instances where there's references associated with this sequence. But there's in this comment field, you can also add notes to this field as you further curate this, sequence. So when you work with a GenBank annotation file, that information is all automatically pulled down as part of that import. Whereas if you're simply adding a sequence using like file new DNA, obviously any notes that you're associating with this file, you would add yourself. So let's just close these two here, since I already had the sequence open in the background. When you initially open the software, there's essentially three parts of the interface. There's the various views here that you can toggle between. So for example, right now I have it in the sequence view. There's a linear map view, circular map views. There's primer design views, primer list views, mini map views, site summary views. We call this down here the feature view. Basically, this will show you all the different features that are a part of that annotation. So if I double-click on one of these features, it will automatically highlight that feature that's present in whatever view that I've clicked on here. As we go through the various views here, there's also a number of folders. So for example, there's folders where if I want to see the bottom strand residues, I can filter by specific enzymes. So if I want to look at all the enzymes in the rebase catalog, I can click on this. I can also sort by features, open reading frames, translations, top strand and bottom strands translations. I can apply annotations in regards to the translation part of it. So for example, if I've identified a specific exon and I just want to create a partial translation specific for, say for example, exon 1 and apply that to my visualization that I'm creating, you also have the ability to simply, if you don't want to turn all the translations on and all the different frames, you can create a translation annotation specific for whatever region of interest you're highlighting. There's also rulers and row information that you can highlight. So essentially you can make this visualization as busy or as simple as you want to do. So what I like to do is I like to manipulate this view a little bit so I can see the sequence view as well as the linear map view and kind of change this a little bit. So to do that, there's a little tab here. So as you can see here, if I hover over this little tab, I can essentially drag this to the right. And then what I'm going to do is I want my features to be up here in the top right-hand corner. And I can click on, I can click on that feature list view. And then down here, I don't want my features list to be just displayed. I actually want excuse me. Okay, I want my linear map to be displayed, or for example, maybe I want my sequence list to be displayed down here in this more of a linear map view. So basically, what you can kind of do is you can change this or manipulate it to whatever is more optimal or for your project, or however you want to set up the visualizations in this interface. So once you manipulate this or set this up to what you like, what you can kind of do is you can also save this either as the default layout template. So what that means is moving forward, every time you open the software, this will be your default view. Or you can also save this as a custom layout template depending on what projects you want to work with in the future. So that is a very helpful tool if you like to manipulate your layout to make it a little bit more customizable for the specific projects that you're working with. So another thing to look at as you work with the software, so in one of the folders that we clicked on was our Enzymes displayed folder. So in the real world, you probably don't want to visualize every single enzyme possible that's present in the Rebase catalog. Obviously, that's going to make your display very busy. What you can kind of do in the software is you can filter by selectors. So if I would expand that little folder that says filter by selectors, what you can kind of do is you can focus on just those selectors. One, maybe that are unique sites or have a specific overhang, or maybe there's a specific vendor that you work with on a regular basis. So we have a number of selectors already built into the software, but you also have the option to create your own selectors based on what you're using in the lab. So to create unique selectors under the enzyme pull down, you can go to our selector manager, or if you automatically just want to create a new selector, basically you can add or delete those selectors that are present in the selector manager. So what I have done previously is I've put in PET39B. I've, from the type pull down, selected pick, and I've just added those enzymes that are present in the multiple cloning site that are unique cutters of PET39B. You can also create selectors based on class, overhang, frequency, pick, or say you want to combine PET39B and Promega Corporation selectors. So there's a number of different selectors that you can create in the software, and then when you apply those selectors and visualize those selectors on the visualization, this is actually an option that you can also save with your default view layout. So I previously applied this, saved this as my default layout template. So anytime I open this project, I'm going to see just those enzymes that are present in the multiple cloning site of PET39B, and then also this specific view that I've set up specifically for this project. Okay. So what I've done here is I've imported JO2846, that tissue factor precursor. So what I'm going to show you do now is what we're going to do is we're going to identify a specific target, an intron in this, and then we're going to start to create primers that will insert this target into that PET39B expression vector. So one thing that I want to turn on automatically is under the features displayed, I'm going to click on features displayed. I already know the frame of this insert, so this target, so I'm going to click on number 1. So this would be the frame that doesn't cause a frame shift, +1 or +2. That would be 2 and 3 here. And I also know it's going to be in the top strand. So I'm going to specifically focus on a specific part of this and a specific open reading frame. This open reading frames exists around 3805. So what I'm going to do here is go in the sequence view, scroll up to 3805, which is right here. Oh, sorry. Right here. So 3805 to 4089, and as you can see, when I highlight in one view, it's highlighted in another, like so. I'm going to go from the priming pull down and select Create Primer Pairs. And what this is going to do is going to open up our primer design dialog that allows you to set specific conditions for salt concentration and also specific primer characteristics. So in this specific case, I'm going to specify, just leave the default primer lengths of 17, maximum of 24. Leave a target TM of 60. You also have the option to set a specific pentameter stability, 3' pentameter stability, as well as uniqueness at the 3' ends. I'm just going to leave these defaults. At any time during the software, while you're in the software, if you have any questions about a specific parameter, so in this specific case, if I didn't know what target TM means, if you click on the Help tool button here, it's automatically going to lead you to the portion in our Help menu, which actually talks about all these different parameters and what it means to change a specific TM and so forth. So definitely want to click on Help if you have any questions about any specific portion of a parameter or a window that you're in. So let's click here, OK. When I click OK, in this primer design dialog at the top, there's a number of different palette tools. So this is just basically a selection mode, allows you to click on specific parts of this sequence view, or I can click on a specific part of the primer, and if I want to make a degeneracy, you can do so. So I could simply click C here, and it would change that A to a C. There's also a palette tool for introducing a restriction site. So we will actually use this to add the SCA1 restriction site and at the 5' end and the XHO1 at the 3' end. There's also a palette tool to introduce a codon change or a mutation. So for example, say, as you can see here, there's a number of false binding sites, which is noted here by those green checks for the top strand primer, and then there's one for that bottom strand primer. So say you want to make a silent mutation that obviously it's not going to affect the translation, but it may minimize that false binding site possibility. You do have the ability here to introduce a codon change and a silent mutation. And there's also a palette tool to quickly navigate between the top strand and the bottom strand primers, like so. Okay. The window that you're seeing here below is it's giving you the most stable dimer pair information. So this would be the propensity of the primer binding to itself. So what you're seeing here is giving some calculations based on the nearest neighbor method. This is actually a combination, or these primers wouldn't be recommended. So the software is telling you it's bad. So it would recommend this specific criteria. So what you would need to do is further optimize this primer to make it not bad and turn it into good. What this kind of showing you here is the possibility for that primer to bind with the top strand, primer to bind with the bottom strand primer. So right now it's giving you it's an okay, but if it would not recommend this combination, this also would say bad. And then here would be the hairpin at the 3' end. Okay. So what we're going to do is simply, I'm going to first add the restriction sites to the 5' and 3' end of these primers. So to do so, I'm going to click on the Introduce Restriction Site scissors. And I know SCA1 is a blunt site, so we're going to insert it right here. I'm going to select any enzyme, and I'm going to simply hit S. And I'm going to scroll down to SCA1. As you can see here, SCA1 is added at the 5' end. It doesn't change the parental strain, the parental nucleotide string. All it does is change the primer here to add that appropriate ends at the 5' end of that primer to add that SCA1 site. I'm going to do so at the 3' end as well. So I'm simply going to click on the Introduce Restriction Site scissors again, go to the 3' end of the target, and I want to simply click X, scroll down to XHO1, and as you can see here, 5' end is my SCA1, and there's the 3' end of my target, which the XH1 has been added. So now the next step is you want to start manipulating this primer to kind of resolve the stable dimer information that's being supplied here, as well as to try to get rid of these false binding sites that exist across this target. So this is actually showing you the entire target, not just the amplified, the highlighted region that the intron that I'm working with. So as you can see here, this is roughly a 13,000 base pair fragment. So it's showing you the possibility where these primers may false bind on this target. So let's first work on the 5' end. Previously, I do know that we want to start with the GCC, so we're going to drag this out to right about here. And then at the 3' end of this target, I'm going to drag this down to the AGAT, which is right about here. And as you can see here, that manipulation has resolved those false binding sites across that template. And then I'm getting some better results in regards to the stable dimer formation, the hairpin information, and the possibility that the primer may bind to the bottom strand. Now let's work on the 3' end. So to resolve this one, I need to drag this out. Let me just take a look here. We're going to drag this down to the GAT, right about here. And then we're going to drag this one out quite a ways to the AGG, like so. So in doing so, in this specific case, we haven't had to add any silent mutations. All we simply had to do is drag 5' or 3' the sequence outward to make it a little bbit more appropriate. So when we insert this amplify this target, essentially it's going to cut here, right at this XHO1 site. So this information at the 5' end of my primer is going to be cut anyway, and then the same thing is going to happen here at the 5' end of my probe. So now what we've done is we've resolved, we have no dimer issues. We don't have any issues in which the primer will bind with, any significant issues with the primer will bind with the bottom strand primer. And then our hairpin information is suitable for, based on the software. The next step is what we've done is we've added those appropriate primers onto the end of our enzymes onto the end of our primers to now we're going to in silico amplify this to create an amplified clone. One question that you may have here is, okay, if you want to save these primers and export those out, on the left-hand side here, we have a folder that says primer pair working set. So expanding any of these, you do also have the option to save as a primer catalog. So I can save it as a .pri file, or I can export this as a FAST or a text file for further downstream use.
Hi, Brian. We do have one question before we go along to discuss cloning.
Okay.
And that question would be, so you've created this primer. Can you use the same primer then in other DNASTAR applications?
Thank you, Jackie. Yeah. So when you export these primer pairs or sets that you're working on, one of those is the primer catalog. So save primer as a primer catalog. So this is actually a primer catalog that you can utilize and run in any application. So we do also have a whole module dedicated to primer design called Primer Select that gives you some more methods and tools to create different reports and so forth. So that is a catalog that you can open in that. Or another tool that's present in our Seq Builder application is I can also import primers from a catalog. So this would be a file, the .pri file, that you could directly import into a sequence. And those sequences, if they hit, would automatically be annotated to the sequence, whether it's a .pri or a FASTA file that you can directly import into a project.
Wonderful. Thank you, Brian.
Okay. So the next step is now what we're going to do is we're going to take these primers, this combination, and start amplifying the ends to be appropriate for insertion into that T7 expression vector. So to do that, what you want to do is you want to click on that 5' SCA1 site. I'm going to click to the 3' end, and then I'm going to highlight the space between by clicking on the XHO1, like so. As you can see here, that whole region is highlighted up to the cut sites of both, like so. So the next step is what I'm going to do from the priming pull down is click Create Insert by PCR. So what I've done here is basically it's automatically created a cloning project. So one of the pull downs from the file, one of the options from the file pull down is the ability to create a new cloning project. For some of the more automated in silico cloning functionality, such as TA cloning, gateway cloning, directional topo cloning, this dialog would automatically be created for you. So when I simply highlighted a region of interest and I go through the Create TA Cloning Insert and the TA Cloning Advisor, it's going to automatically place the target, the vector that you use for that expression, and then the resulting clone where that target has been inserted into the expression vector. So we're essentially doing the same thing here in the project. So we've amplified the ends of this target to be suitable for expression into the that T7 expression vector. So to really take a look at this, we can close this one and actually just take a look at that insert. As you can see here, my view has been retained. So there's my SCA1 site at the 5' end. There's my XHO1 site at the 3' end. When I click on that, as you can see, there's my ATG. There's my initiating methionine. There's my ACT, which is residual from the SCA1. There's my initiating methionine, ATG. And then the same thing, the cut site for XHO1 exists at the 3' end. One thing that I like to do to kind of look at and make sure everything is in frame is under the Format pull down, you do have the option to specify a type of grouping. So in this specific case, group by 3, and that will definitely help with looking at the frame of this insert. One thing you'll notice here, since I add some of those residual, those primers towards the end of my sequence, there is a frame shift, it looks like here, but since I'm going to use the SCA1, so the SCA1 cuts between the T and the A here, my ATG is going to be maintained. So in this specific case, ACT be the 3'. So if you wanted to get everything in frame, you can add, residual ends here to get everything in frame. Like so, as you can see here, I've added an N. There's my ACT, and there's my ATG. And then also I can apply, the frame, the open reading frame. That's what this visualization here is showing you, where my initiating methionine, where it's going to translate, and then my termination there at the 3' end. So that's one thing that I kind of do just for a visualizations perspective is I like to change the formatting specifically to group by 3 so I can actually see where that initiating methionine is and make sure everything is framed with the upstream elements, the promoters, and so forth. Another thing you can do here is, if you have a question about what specifically this open reading frame is, we already kind of know that this is associated with that tissue factor precursor, but saying you're coming from a cloning project, you've identified a large sequence on a gel, and you have no idea what the function is. There's also built into the software the ability to blast the selection. So I can highlight that open reading frame and blast that selection directly to various databases that are present in the software very easily, and visualize and see what's the function of this open reading frame and so forth. So those are a couple things you kind of do to take a look at this amplified target to make sure everything is in frames. You can also blast by functionality. The next step is we're going to take this target and insert it into that T7 expression vector. So I'm going to click on that SCA1 5' site again. Click on that XHO1 site to highlight the whole region between. The next step is I'm going to go to the cloning pull down, select copy restriction insert. What that essentially is doing is copying it to your computer. And now we want to focus on that APET 39 expression vector. So since this expression vector is coming from a company that sells this as a commercial product, you can, and specifically working with the enzymes within the multiple cloning site, the frame is pretty much going to, it's part of their QC process. They make sure that any restriction site they recommend that exists in the multiple cloning site is going to be in frame of that T7 promoter and initiating methionine that exists. If you're working outside the multiple cloning site, it's going to be more important to verify, make sure that everything is in frame, whether with a promoter that you're inserting specifically or simply within in frame of that promoter that's being used for expression. So what I'm going to do here is, I know the multiple cloning site exists at the 3' end of this vector. I'm going to turn the features on, and then I'm simply going to zoom in on that multiple cloning site. So I'm going to click on the selection mode selector. We're going to click on the SA1 site, the corresponding sites that we've clicked on in the target. And then to complete this, we're going to go to Cloning, Clone Restriction Insert. As you can see here, a dialog box pops up that shows me the left end, or the 3' end of my vector, in the 5' end of my insert. So it's showing you, okay, does that AGT, ACT site match up? It's also showing you the right end of my insert in the 5' end of my vector, so it's showing you that XHO1 site. So If for some reason your selection doesn't match up, one of these will be checked, unmarked, not checked. So in this specific case, it's saying this selection you've identified, there's appropriate sites on my target that have the SCA1 and the XHO1, and there's appropriate sites in my vector that have the SCA1 and the XHO1 sites. Also in this window, it's kind of showing you, okay, what is the frame? So it's recommending in this specific case, the reading frame is 1. If for some reason you have a frame shift in your vector or in your target, it may recommend another frame here, so frame 2 or 3, depending on what it gives. It gives you an option to change these two if you also want to specify a different frame. Also, if you want to do the reverse complement, you can check the reverse complement. But to complete this insert into this expression vector, basically the next step is you would click clone. What the software does now is it has created a untitled clone. So if we go back to that cloning project here, as you can see here, here's my amplified insert. There's my clone, untitled clone, that has been inserted into that clones folder. And then there's my untitled clone that I can further evaluate and take a look at. So when looking at this clone, a couple things that I like to look at is, one, I want to make sure all the features are on or displayed. So the features are on, displayed. I also want to take a look at the open reading frame, so I know it's this is the frame, number 1. But I also want to take a look at, what's being shown. So there's that, that tag that's been inserted at the 5' end of that target. So you can kind of start taking a look at, okay, there's my initiating methionine. There's my T7 promoter. You can kind of take a look at the spacing between that initiating methionine and that T7 promoter just to make sure there's sufficient room, like so. If I drag this up, you can also start taking a look at, okay, there's where the initiating methionine is starting. It's going to translate my DSBA tag. It's going to go through my target that I've added, specific going through the XHO1, and then there's my terminator at the 3' end. So simply looking at the frame, the open reading frame, that's kind of what this little checkbox here. A solid triangle identifies that yes, there's an ATG, there's initiating methionine. This is going to be translating through my target of interest and then terminating at the terminator that exists in the PET39B expression vector. So looking at this, I've identified that the frame appears okay, and now the next logical steps is I can start translating this.
Hey, Brian, before we go on to the translation, we do have a couple questions here. The first of which, so we see all these features here in Seek Builder. Is there any way we can customize these features a little bit, change the color, something like that?
Sure. Everything that you see here, you can manipulate. So we can, we've changed the visualization, how this visualization is being shown. But you can also change specific elements or features that exist in the representation. So like I said previously, these are all annotations that previously exist in the file that I brought down from NCBI or the PET39B plasmid that I've already brought into the software. So these have already been added. But say you have your own sequence that you're bringing in that has no annotations, that is simply from a Word document. From the File New pull-down, you have the option to also add your own specific nucleotide string, and you have the ability to annotate that information. Any feature that you've created, you have the ability to further edit what's being displayed. So, for example, if you don't want this is actually a miscellaneous feature. You can start identifying, okay, these are all standard GenBank nomenclature. So for in this specific case, I want to say that this is a RBS, like so. There's a default color that exists for that RBS. And I can name that RBS. So I can call that Trix or whatever. And as you can see here, when you name that, it's automatically added to the feature. So you can add whatever you want feature-wise as well as nucleotide or protein string-wise in Seek Builder, and then you can annotate it as well accordingly. So let's actually go back to the previous project. So to say you wanted to change the style of this specific feature here, like so. What you would simply do is click on it, and it would essentially highlight that feature. Right-click on it, and then select Edit Feature Style. So as you can see here, there's a default style, color, fill, shadow, as well as shape that exists with this specific feature. So if you want to individually change this feature, I can change it to something else. So maybe I want it a yellow color. Apply that, and that yellow color will be automatically edited for that individual feature. While I'm in the linear view, as you can see, I can actually drag these and change the manipulation where you want this to be shown. You can also detach feature labels. This is editable. Another thing, what I call a global feature change. In this Features Displayed folder, you can sort by alphabetical list, positional list, as well as feature types. So if I simply expand on feature types, say you want all your CDSs to be a specific color or style, if I right-click on the CDSs, I can also change what I call the global default feature style. So in this specific case, if I want all my CDSs to be green, select OK and apply, all my CDSs in this specific project will be changed to a green coloring or that specific feature style that you've identified in that the shape arrow here. So it's a filled arrow, for example. If you want to make this your default layout change, so you can manipulate these, change the coloring styles based on whatever you prefer, you would simply go to the View pull-down, and then I can save that layout as my default template. So moving forward, any color changes or visualization changes that you make here, that's going to be retained in this project as well as every project that you work with moving forward.
Great, thanks, Brian. One other question before we move on. So we've created this expression clone here. Is there any way we can use this expression clone we've created in a publication?
Sure, yes. Any of the visualizations that you see here, you do have the option to, I can highlight a selection and edit, copy that selection as a picture, or there's some options here to copy it as a translated sequence or an unformatted sequence. So if I copy as a picture, I can easily basically insert this into a PowerPoint or Adobe Illustrator or Microsoft Office or Word. These are vector images that you can edit further in those other applications. So not just pasting the actual TIFF or the PNG, the image, it actually is an image that you can further manipulate or edit directly in Word processing document.
Great, thanks, Brian.
Okay. All right, so what we've done here is we have our we have our clone. Our clone has been inserted in a PET 39B. So the next logical steps is now I want to take a look at the translation of this. So why would I want to look at the translation of this? So when you're trying to purify this, maybe it's in the insoluble fraction. With this strategy, using this DSBA tag, there's a high likelihood that since it's being forced to the periplasmic space, that it will be in the soluble fraction, but maybe it's in the insoluble fraction. A couple things as based on your primers that you've designed, maybe that binding site of your target is embedded in the actual, when you have the DSBA tag added to the 5' end of your target, maybe the actual primer site may be embedded within the protein. So these are all things to think about when you're doing your analyses and you're using your target-specific primers, that if they're not being recognized or not amplifying something, it could be simply embedded with that DSBA tag. So what I kind of like to do here now is you can translate with that DSBA tag that's been attached, or if you simply want to translate your target. So what I'm going to simply do here is just do a translation with the DSBA tag that has been added to the 5' end. And to simply do that translation, you would simply go to the sequence pull down, select new sequence, and translate. What the software is kind of showing you here, click on the comment view here because these are some DNA statistics that are very helpful for taking a look at this protein. All this basically has done is there's my initiating methionine, it's translated through, and there's my termination at the 3' end of the sequence. This information is very helpful because it's going to give you your molecular weight, so when you run this on a gel, it's going to show you what's the approximate size of your fragment that you should expect. So 37 kilodaltons here is the expected fragment size. It's going to give you some amino acid breakdowns. It's going to give you a charge at pH 7.0, your isoelectric points. your AAGTC breakdown. And then one thing that's very helpful, this is a human protein. Since we're trying to express this in E. coli, it's actually going to show you a codon usage chart table here down at the bottom. So for those of you who are not that familiar with rare codons are basically lower frequency codons that exist in different organisms. So for example, since we express this in E. coli, some common arginine rare codons would be AGG, AGA, leucine would be CUA, proline, CCC. So simply looking at this, I have some arginines here that there is some rare codons that are present. So there's 3 here, 2 here. So as you further evaluate what you're seeing on the gel, maybe you see some truncation, and you can't figure out why that's occurring. Well, in this specific case, 5 arginines in a sequence is not overtly significant a lot, but typically 8 to 9 rare codons, there's a possibility that it may affect your translation, truncated results, low expression, and so forth. So depending on what you're seeing here, you may want to think about using a competent cell that has some tRNAs for these rare codons to get a more efficient expression. So for example, EMD Millipore has the Rosetta series of strains and so forth. So this is a very helpful interface or a translation results where I can kind of take a look at and really look into what I'm translating, what I'm seeing, what's my expected result on a gel, as well as if I'm seeing something interesting, not expected on that gel that I can kind of further extrapolate, okay, and maybe it's a rare codon issue. So the next thing that I kind of try to look at is we've started taking a look at some of the DNA statistics. So what we're expecting to see on the gel. The next couple of things that we can kind of take a look at would be in our Protein 3D application. So I'm simply going to launch that Protein 3D tool. So Protein 3D is a newer tool that's been added to our suite. So it is a tool that I can bring in protein data bank files. So in this specific case, there's a tissue factor PDB file that's been crystallized, that's been submitted to the protein data bank, and then simply, I can visualize that, zoom in and zoom out of this, on this sequence. There's a number of different methods that I can apply to this 3-dimensional visualizations to assess for, disorder, flexibility, hydropathy, secondary structure, transmembrane, antigenicity, B cell epitope prediction, so forth. There's a number of different physiochemical methods you can apply this protein to further extrapolate. Okay, what am I seeing? What are these specific areas, and what's the physiochemical significance? There's also cleavage sites, so I can apply proteases. There's also prosite information. I can apply prosite domains and sites information. And then one other very helpful tool is, there's a direct link to the Yale Morph server. So there's a number of different molecular motions in a motion library where what Yale, the Morph server, has done is actually taken a number of different crystal structures and actually animated them. So you can see these proteins opening and closing, like so. Let's just, if I click on in the background, you see this protein like so, opening and closing. So if you're working with some of those proteins that are present in that database, it's very helpful to try to see what's the interaction, how the domains are opening and closing for you. For a cloning project, one thing that may be helpful, like I previously mentioned, is not everything is going to be crystallized, but what we've added to the Protein 3D application is actually our novel fold interface. So for example, I've previously brought in that target protein, so simply by selecting File, Open, and then navigated to that protein that was called target.pro. So say, for example, when you have applied some of these methods, maybe there's a Kite Doolittle or a Hopwood's method for hydropathy that's kind of showing you based on the amino acid information, what is how hydrophobic or how hydrophilic is that specific region of interest? So what these methods are showing here, so if I simply highlight this little portion here in the hydrophobicity plot, in this specific case, there's a scale of -4.5 to +4.5. This specific part of the amino acid or the target is hydrophobic. So this may be something that's in the transmembrane or the intracellular space. But what you can kind of do is, as you highlight these selections, I can basically utilize our NovaFold application to create visualization. So to verify that, I can highlight that selection and select structure, predict structures, and create a prediction for the highlighted or the selected region. Right now, the 400 amino acids would be the largest. We're expanding on that here in the near future. You can also align specific structures together. So say you have a SNP that exists in a known PDB, but as you fold the new target that it has the incorporation of the SNPs, you can align those two SNPs together, those two proteins together, and actually visualize the interaction in the three-dimensional space where those SNPs exist. Or in this specific case, I can simply highlight that selection and align it. So, for example, 28 very small residues, I've highlighted it, and I have generated a three-dimensional image of that selection. So that's roughly took like 3 hours. It's kind of showing you the three-dimensional representation. So this will allow you to extrapolate. Okay, what's the is this a is this a hydrophilic protein? Where does it exist? What does it look like in the three-dimensional space? If you have a specific focused area of interest, where that exists in that three-dimensional image, and so forth. Directly within the software, there's a number of different visualizations that you can kind of change, so style patterns. So if you want to change your side chains to be a specific visualization like spheres or so forth, or you want a specific coloring style, you do have the ability to manipulate this image, and then further, I can save this image as an image file that I can use for a presentation or a PowerPoint or a publication for further downstream analysis. Basically, when analyzing the protein, like I said, the end result of if you've discovered an enzyme or a protein of interest that does something unique, the end result typically is you're going to crystallize it so you can kind of take a look at the three-dimensional image. So what we've done here is we've incorporated a tool that allows you to do that, create a three-dimensional image, but it utilizes a very accurate algorithm that will do that without having to go do all those steps that would crystallize a structure. So those are some of the logical steps that you could think about when you're working with a cloning, expression, and primer design type of project. And so to summarize, basically what we've done is we've PCR amplified a target. We've inserted that target into a T7 expression vector. We've analyzed that expression vector using a number of different methods, and then we've also analyzed the translated protein for further downstream analysis. That concludes the overview for today. I hope it was helpful.
Thanks, Brian. We do have one last question before we wrap up. So you should NovaFold a little bit here in this presentation. And so if we kind of look at the bottom of your screen, we had someone asking about that NovaFold license message there. Now, if they already have Protein 3D, is that something that should automatically pop up for them?
Sure, that's a great question. So basically, Protein 3D, the NovaFold application within Protein 3D is actually a tool that's run via the Amazon Web Services. So this is a tool that we have a number of different pricing options. So for some labs, they're working with one enzyme, one protein, and that's all they were going to focus on, and the end result is one crystallization. So we do have some different pricing options that if you only want one prediction, and since there is a cost to manage and run that Amazon Web Services, there's a number of different pricing options. Whether it's one prediction, you can set it up by you pay a certain amount a month, unlimited for a month, or a year options. But yeah, this is actually a tool that doesn't automatically work with Protein 3D. It is a cost there is a cost to running the NovaFold application because it does run via the Amazon Web Services.
Great, thanks, Brian. So we have a couple questions left, but they're more highly technical questions, so I'm going to follow up with those people personally after the webinar. If you think of any other questions later, you can email those to me at webinars@dnastar.com, or again, tweet us at DNASTAR Inc. Also feel free to email me with any ideas for future webinar topics that you'd like to see us present. So thank you again for joining us today. We hope you found this webinar helpful. We do have a large collection of videos on our website, as Brian mentioned earlier, so I encourage you to check those out when you have some time. Our next webinar is on Thursday, August 22nd, where we will be discussing using prostate cancer exome data to identify candidate genes at the Genetic and Genomics BioConference Live events. After that, on Wednesday, August 28th, we will also be presenting an advanced SNP analysis webinar. You can watch the recording of any of our past webinars as well as register for these upcoming webinars right here on our webinars page. We hope to see you there, and thank you again for joining us today.